Salt marshes are intertidal marine ecosystems that form in areas of reduced oceanic flow, such as in bays and inlets. During periods of low tide, they are exposed to the air, while during high tide they are submerged in salt water. This daily flooding and draining of salt marshes has caused salt marsh plants and animals to become adapted to the challenges of both marine and terrestrial environments. Unlike many terrestrial ecosystems, such as forests, where sediment is geologically deposited or eroded from underlying rocks, salt marshes are built biologically by the organisms that inhabit them. Vascular plants colonize inlets and bays, and over time physically trap sediments from both oceanic and freshwater sources. These plants bind sediments together, and in combination with their decaying below-ground plant matter, slowly build a complex substrate called peat that forms the base of the marsh environment. On the east coast of the United States, where salt marshes are common, the plant responsible for building salt marsh habitat is Spartina alterniflora. Spartina, or cordgrass, initially colonizes the shores of inlets and bays, and once a salt marsh begins to form, it remains on the outer edge of the marsh, constantly binding sediments and pushing the edge of the salt marsh outwards into the ocean. As salt marshes extend further from the coast, wave energy increases, leading to increased erosion at the leading edge of the marsh. Salt marshes are constantly expanding and eroding, creating dynamic and interesting ecosystems. Unfortunately, salt marshes in the United States are seriously threatened by human activity. Historically, dredging and marsh development destroyed and transformed salt marsh habitat, while ditching for mosquito control during the Great Depression altered the hydrology and biology of salt marshes. These threats still persist and are accompanied by new stressors, such as increased nitrogen loads, sea level rise, and pollution. Another threat to salt marshes that remained underappreciated for decades is recreational overfishing. Overfishing has caused catastrophic ecological damage in many marine ecosystems, such as kelp forests and coral reefs. Overfishing affects marine ecosystems by removing top predators, which has a trickle-down effect on the rest of the ecosystem, known as a trophic cascade. A trophic cascade is an ecological phenomenon in which one trophic level, one level in a food chain, undergoes population increases or decreases, which have cascading effects down the food chain. In New England salt marshes, a trophic cascade occurs when populations of predator species, such as striped bass and blue crabs, are greatly reduced due to recreational overfishing. These species play important roles in salt marsh ecosystems, which the Bertness Lab has documented over the last 4 years. These species help maintain and control the populations of many species, including the nocturnal herbivorous burrowing crab, Sisarma reticulatum. Sisarma is a native species, but is typically found at very low numbers in healthy marshes where its predators are abundant. When these predators are removed by recreational overfishing, Sisarma populations explode, increasing up to 400% in the most heavily fished marshes. These increased densities have serious effects further down the food chain. Sisarma are herbivorous crabs and graze almost exclusively on Spartina alterniflora, the cordgrass on the leading edge of New England salt marshes, responsible for binding and trapping sediments. Sisarma grazes on cordgrass leaves above ground and creates complex burrow systems that allow them to feed on cordgrass roots and rhizomes without fear of predation or heat stress. Sisarma grazing and burrowing creates large stretches of bare creek bank with no above-ground or below-ground vegetation, and large Swiss cheese-like burrow complexes. Without cordgrass to hold the substrate together and bind new sediment, serious erosion occurs, and the leading edge of the marsh can retreat several feet per year. The marsh erodes vertically and horizontally, and when large chunks of peat become structurally weakened, they split off into the ocean. This leads to very significant habitat loss at the marsh edge, where marshes are built and where the effects of sea level rise are most strongly felt. When entire swaths of peat are lost, what was once marsh becomes large patches of soft, muddy sediment. On Cape Cod, where the Bertness Lab has done the bulk of its recent research, salt marsh die-off has been occurring since the late 1970s. In the last few years, however, we've seen some interesting examples of cordgrass recovery at die-off marshes. Some grazing by Sisarma is still occurring, but large patches of bare substrate at the marsh edge have become revegetated. The regrowth we're seeing, though, is not in the bare, heavily eroded peat that remains after Susarma grazing and burrowing occurs, but in the soft, muddy sediment that occurs below the eroded peat. The cordgrass regrowth forms long bands parallel to the marsh bank and typically grows right to the border of the heavily eroded peat. The cause for this partial recovery is still not entirely known, but we are currently investigating several possible hypotheses. It might be caused by an increase in Susarma predators, which would reduce Susarma populations and decrease the amount of grazing on cordgrass. It might also be because cordgrass is regrowing into a zone where Susarma cannot graze it. While the lab works on these larger questions, I have been trying to understand the effects and importance of this regrowth. So far, I focused on how cordgrass recovery changes the soft, muddy sediment in which it occurs, and what the future trajectory of salt marsh recovery may be. I've also investigated whether regrowth rebuilds marsh habitat in the same manner that normal cordgrass promotes marsh construction over many centuries, and if regrowth creates a positive feedback cycle that promotes increased cordgrass growth and expansion into new areas. We have developed both field and lab experiments to help us answer these questions related to the importance and trajectory of salt marsh recovery. The first experiment was set up in the field at a marsh adjacent to Lewis Pond in West Yarmouth, Cape Cod. The goal of the experiment was to determine the effects of regrowth on the marsh landscape. More specifically, we wanted to know whether cordgrass regrowth collects and binds sediments and converts it into marsh habitat on a short timescale. To do this, we manipulated existing patches of regrowth by eliminating all vegetation in 10 1x3 meter plots of regrowth. Using permanent PVC pipes, which we embedded into the substrate, we then quantified changes in surface elevation over time to see if the loss of vegetation would lead to net marsh loss. We expected the removal of vegetation to destabilize the substrate and lead to rapid erosion, since we know that cordgrass is necessary to combat the eroding effects of waves and ocean currents at the leading edge of the marsh. We compared these mowed-down plots to similarly sized plots of normal, unmanipulated regrowth, and to plots of muddy, soft sediment that had not experienced regrowth of any sort. The results of this experiment showed definitively that the presence of vegetation has a significant effect on sediment dynamics. The experimentally mowed-down plots decreased 1.5 centimeters in surface elevation after only 3 months, while the plots with cordgrass regrowth had no significant net change in surface elevation. Interestingly, the muddy, soft sediment plots without regrowth decreased by around 0.8 centimeters. These results suggest that vegetated creek banks are crucial for marshes to fight the effects of erosion and sea level rise, and that cordgrass that has regrown at die-off sites is capable of providing the service to marshes at risk of future erosion. The next step involved setting up a greenhouse experiment to look at whether sediment from areas with regrowth and areas without regrowth had different effects on cordgrass growth. We collected these two types of sediments, one from areas within regrowth vegetation and the other from areas that didn't have any cordgrass regrowth, and used them to fill small flower pots. We then planted a single cordgrass seedling in each pot to look for differences in growth in the two sediment types. We took various measurements, including seedling height and above and below ground biomass. Cordgrass spreads primarily asexually by sending out below ground runners that become full-fledged plants. We also took measurements on the number and heights of asexually produced plants each seedling generated. This experiment showed that sediment type had a significant effect on seedling growth. First, we saw a significant difference in below ground biomass. Seedlings grown in sediment from regrowth areas produce significantly more below-ground biomass than seedlings grown in sediment from unvegetated, muddy sediment. Below-ground biomass is important because it contributes to marsh stabilization and helps fight erosion. We also saw a significant difference in reproductive output. Seedlings grown in sediment from regrowth areas sent out more asexual runners and had a greater total height of runners than seedlings grown in sediment from areas without regrowth. This means that seedlings grown in sediment from regrowth areas produce a greater number of asexual offspring than seedlings grown in sediment without regrowth. These two results provide evidence for what scientists call a positive feedback. The initial growth of vegetation into formerly unvegetated muddy sediment changes the sediment in some way that facilitates further regrowth, creating a positive cycle of plant growth. This means that the leading edge of marshes that have regrown cordgrass will continue to increasingly bind sediment and resist erosion, and should continue to expand marsh habitat into the future. Salt marshes are critically important ecosystems for humans and provide a variety of services to surrounding human populations, including coastal flood protection, nutrient filtration from sewer and septic runoff, and as nursery grounds for a great number of commercially important fish and invertebrate species. The loss of salt marsh habitat that we have documented over the last 4 years has led to reduced coastal flood protection, increased nutrient runoff into estuaries, and reduced sustainability for several important fisheries. For all these reasons, salt marshes are amongst the most valuable ecosystems we have in the Northeast, especially in areas like Cape Cod and Long Island, where flooding and sea level rise are very serious threats, and local economies depend upon recreational and commercial fishing. As a result, it is important not only to protect salt marshes from future harm, but also to develop an understanding of their resilience and potential to recover from disturbances like salt marsh die-off. Salt marshes have a long history of abuse at the hands of humans, but our recent work leads us to believe that New England salt marshes may have the ability to recover from this most recent impact, salt marsh die-off. When the Bertness Lab began investigating the phenomenon of die-off on Cape Cod, we saw very few evident signs of recovery and had little hope salt marshes would recover in the foreseeable future. With the loss of Spartina alterniflora, or cordgrass, from the seaward border of the marsh, and the resulting loss of critical cordgrass habitat, the continued negative effects of die-off seemed inevitable. The recovery we've seen in the past few years, though, has led us to believe that these ecosystems may be more resilient than previously imagined. If we do not understand the mechanism for this recovery, though, or understand how important this recovery may be for marsh restoration, we will not be able to make effective conservation decisions concerning salt marsh health. In the future, we will continue to improve our understanding of why this recovery occurred, and more importantly, how we can promote more widespread recovery across Cape Cod. The work I have done in the Bertness Lab represents an important step towards understanding how salt marshes respond to die-off, and will inform how we prevent similar situations from occurring in the future.